Welcome back to my channel Only Bio. In this session, I have taken up the NEET question paper of 2020 and have solved the MCQs along with the explanation. So I've divided it into 9 parts. So this is the first one, that's 1 to 10. So we begin with this. The first question relates with the chapter genes, and the question is, if the distance between two consecutive base pairs is 0.34 nanometers and the total number of base pairs of a DNA double helix in a typical mammalian cell is 6.6 into 10 raised to 9 base pairs, then the length of the DNA is approximately dash. And the options given are A. 2.5 meters, B. 2.2 meters, C. 2.7 meters, and D. 2 meters, and the correct answer for this question is B, that is 2.2 meters. Now the distance given between two base pairs is 0.34 nanometers, right? So this is going to be equal to 0.34 into 10 raised to minus 9 meters. Same you have to apply it for the total length of the DNA given. So this length, as we can see, is far greater than the dimension of a typical nucleus, which is approximately 10 raised to minus 6 meters. So this is how we calculate. It's going to be 6.6 into 0.34. Okay, and this is how the answer comes to 2.2 meters. The next question relates with the chapter classification of animals, and the question is, bilaterally symmetrical and acoelomate animals are exemplified by dash. The options are A. Platyhelminthes, B. Achyhelminthes, C. Annelida, and D. Ctenophora. And the correct answer for this question is A, that is Platyhelminthes. We can take an example of liver fluke, right? So you can imagine that. These are the first acoelomate animals. Acoelomate is without a coelom and which have organ system organization along with the bilateral symmetry. The next question relates with the chapter classification of animals, and the question is, match the following columns and select the correct option. We have two columns, column 1, column 2, and there are A, B, C, D options given there. And the options given here are this. The correct answer for this would be A, that is gregarious polyphagous pest is locust, a locusta. B, adult with radial symmetry and larva with bilateral symmetry is going to be Asterias. C, book lungs is going to be scorpion. And D, bioluminescence is going to be Tinoplana. Now let us understand this. Friends, gregarious means animals living in flocks to form the colonies or groups. Polyphagous means able to feed on various kinds of foods. Locust jo hote, okay, locust, actually this word locust is derived from the Latin word locusta, which means grasshopper. So these are a collection of certain species of short-horned grasshoppers, you may say. So these grasshoppers are normally innocuous, means they're not harmful. The numbers are low and they do not pose a major economic threat to the agriculture. However, under certain conditions of drought, which are followed by rapid vegetation or growth, what happens is there's a release of serotonin in their brains. And this triggers a dramatic set of changes. They start to breed abundantly, becoming gregarious and nomadic. So they are something like migratory, you may say. And when their populations become dense enough, they form the bands of wingless nymphs and later become swarms of winged adults. And both the hands and the swarms, both the bands and the swarms move around and rapidly they strip the fields and cause the damage to the crops. Second is Asterias, which is a genus of Asteridae, a family of sea stars. It includes several of the best known species of the sea stars like starfish, Asteria rubens, Asterias amurensis. And these species have planktonic larvae. Okay. Now, the third option is book lungs, which are a form of respiratory organ. You can look on the screen for understanding what a book lung looks like. Okay, so these are found in certain air-breathing arachnid arthropods like scorpions and some spiders. Each book lung consists of a series of thin plates. You can see this very clearly in the picture. These are highly vascular. That means they are richly supplied with blood and are arranged in relation to each other like the pages of the book. These plates extend into an internal pouch formed by the external skeleton that opens to the exterior by a small slit, and this provides an extensive surface for the exchange of oxygen and carbon dioxide with the surrounding air. Now, another thing that can be kept in mind here is scorpions have 4 pairs and spiders have 2 pairs of these book lungs. Now, let's understand the fourth option, that's bioluminescence. Look on the screen, please. Bioluminescence is the property of the living organism to emit light. You can say it is a kind of chemiluminescence because it involves a chemical reaction which involves some light emitting molecule called luciferin and an enzyme called as luciferase. Now, bioluminescence is mostly seen in marine vertebrates and invertebrate animals, in few of them, of course, in few fungi, few bacteria, and terrestrial invertebrates like the fireflies. Now, Tinoplana, which you can see on the screen, is a genus of comb jellies, which are transparent or colorless organisms and belong to the phylum Ctenophora. They exhibit the property of bioluminescence, which is caused by the activation of calcium. So, calcium activated proteins, pehla calcium, this in turn will activate the proteins named as the photoproteins in the cells called as the photocytes in these organisms. The next question relates with structural organization of the cell, and the question is, which is the important site of formation of glycoproteins and glycolipids in eukaryotic cells? And the options given are peroxisomes, B. Golgi body, C. Polysomes, and D. Endoplasmic reticulum. And the correct answer is Golgi body. Now, friends, peroxisome, the first option given, it is a membrane-bound organelle which was earlier called as a microbody, which is found in the cytoplasm of virtually all eukaryotic cells. Now, these are oxidative organelles. Frequently, molecular oxygen, it may serve as a core substrate from which hydrogen peroxide is formed. Peroxisomes hence are called so because they owe their name to hydrogen peroxide generating and scavenging activities. So they perform a key role in lipid metabolism, are involved in the catabolism of very long chain fatty acids, branch chain fatty acids, bile acid intermediates, and certain amino acids in hydrogen peroxide. Now let's talk about Golgi apparatus, which is also known as Golgi complex or Golgi body. And this is a membrane-bound cell organelle found in eukaryotic cells. It is made up of a series of flattened stacked pouches like cisternae. It's a part of the endomembrane system in the cytoplasm, and that is the reason why it's located next to the endoplasmic reticulum and also near the cell nucleus. It is responsible for transporting, modifying and packaging proteins and lipids into vesicles for delivery to the targeted destinations. You know, its function can be related with that of a post office because proteins are modified, sorted and packaged by the Golgi apparatus. In general, it helps in absorption of compounds, formation of secondary vesicles and secretion, formation of enzymes and hormones, formation of acrosome, formation of intracellular crystals, and so on. They are also involved in the transport of lipids around the cell and formation of lysosomes. Now, the option C is what are polysomes? These are nothing but a cluster of ribosomes. They are held together by a strand of mRNA that helps in translation process. Now, what is D? That is endoplasmic reticulum. That's a network of membranous tubules within the cytoplasm of a eukaryotic cell, continuous with the nuclear membrane, and it usually has ribosomes attached and is involved in protein synthesis. Now the next question relates with the chapter circulation. The question is, the QRS complex in a standard ECG represents dash, and the options are A. Depolarization of the auricles, B. Depolarization of the ventricles, C. Repolarization of the ventricles, and D. Depolarization of the auricles. And the correct option is B. Depolarization of the ventricles. Let's look on the screen here to understand the ECG. You can see clearly the standard ECG showing up PQRST, the waves. Either you call it as ECG or EKG. Depolarization occurs in four chambers of the heart, both atria first and then both ventricles. The SA node on the wall of the right atrium initiates depolarization. Here you can simply remember it as depolarization is nothing but the contraction in the right and the left auricle, causing this forceful contraction which corresponds to the P wave on an ECG. Now, SA node sends the depolarization wave to the AV node, that is the atrioventricular node, with which is about about 100 milliseconds delay pe hota hai after the SA node, you can say, to let the atria finish contracting and then causes contraction, that is depolarization in both the ventricles, and this is seen in the QRS wave. So at the same time, the atria repolarize and relax. The ventricles are repolarized and relaxed in the next, that is the T wave. So this process continues regularly unless there's a problem in the heart. The next question relates with the chapter human skeleton, and the question is, match the following with the correct options. You have two columns, column A and column B, and you have been given the options. The correct answer for this is going to be 3. That is floating ribs. You can relate this up to D. That means A is D, that is the fourth one. A is the fourth one. Do not connect with the sternum. Acromion is the head of the acromion is going to be at the acromion process. That's at the clavicle region, right? So it's third one. Then C, that is scapula, is located between the second and the seventh rib. So that's the first. And glenoid cavity, that is going to be something which connects with the head of the humerus. Now look on the screen. You'll get an idea for what is meant by floating ribs. One of the last two ribs, a rib is said to be floating if it does not attach to the sternum, which is the breastbone, and also it doesn't attach to the other rib. There are usually 12 pairs of ribs in all. Each pair of ribs is attached to the building blocks of the spine, that is the vertebrae in the back. The 12 ribs can be classified as the true ribs, that is the first 7 pairs of ribs which attach to the sternum and hence are either known as true ribs or vertebrosternal ribs. Then the lower 5 ribs which do not directly connect to the sternum are known as the false ribs. The upper 3 false ribs connect to the costal cartilage of the ribs just above them, and the last 2 false ribs in a real sense, however, usually have no ventral attachment. So they are called as the floating ribs, also at times called as the fluctuating or vertebral ribs. Look on the screen for understanding the acromion process, the scapula. Okay, the acromion is a bony process on the scapula. Together with the coracoid process, it extends laterally over the shoulder joint. It is a continuation of the scapular spine and hooks over anteriorly. It articulates with the clavicle to form acromioclavicular joint. The scapula, plural is scapulae or scapulas, which is even called as the shoulder bone, shoulder blade, wing bone or blade bone. It is the one that connects the humerus, that is the upper arm bone, with the clavicle. Like their connected bones, the scapulae are paired with each scapula on either side of the body being roughly a mirror image of the other. So the scapula forms the back of the shoulder girdle. In humans, it is a flat bone roughly triangular in shape placed on posterolateral aspect of the thoracic cage. The glenoid cavity, which is also called as the glenoid fossa, it's a part of the shoulder. It is a shallow articular surface which is located on the lateral angle of the scapula, which articulates with the head of the humerus. This cavity forms the glenohumeral joint. I repeat, it is glenohumeral joint which is classified as ball and socket joint. So यहाँ पर आप याद रख सकते हो कि ball and socket joint over here can be even termed as glenohumeral joint. The next question relates with the chapter chromosomal basis of inheritance, and the question is, experimental verification of the chromosomal theory of inheritance was done by dash, and the options given are A. Sutton, B. Boveri, C. Morgan, D. Mendel, and the correct answer is C. Now friends, remember Sutton and Boveri proposed a chromosomal theory of inheritance, whereas Morgan, the full name being Thomas Hunt Morgan, he studied fruit flies, that is Drosophila melanogaster, and provided the first strong confirmation of the chromosome theory. He discovered a mutation that affected the eye color of Drosophila, and he observed that mutation was inherited differently by male and female flies. And based on this inheritance pattern, he concluded that the gene responsible for the eye color is located on the X chromosome. The next question relates with the chapter morphology of plants, and the question is, identify the incorrect statement, and the options given are A. Sapwood is involved in conduction of water and minerals from the root to the leaf. B. Sapwood is the innermost secondary xylem and is lighter in color. C. Due to deposition of tannins, resins, oils, etc., heartwood is dark in color. D. Heartwood does not conduct water but gives mechanical support. And the incorrect statement among these options is B. Sapwood is the innermost secondary xylem. That's a wrong statement. Let us understand why. Look on the screen here. Sapwood is the outer layer of a branch or a limb that is still living, and you can very clearly see it is lighter in color and is very moist. It is the living part of a tree where the sap and the water flow. That is the reason why we call it as sapwood. All wood initially grows as sapwood, but as the tree ages, the innermost layers of the sapwood turn to heartwood, which is no longer living and does not transport nutrients. Sapwood usually has a lighter color than the heartwood that it surrounds. It is high in moisture due to the steady movement of sap and water. Sapwood is not ideal for many woodworking projects due to its high moisture content. This causes the wood to shrink as it dries, and it also makes the wood more susceptible to decay and fungus. However, it is used in small projects. Now, in woody trees, the central portion of the stem is always dark, right? Because this is hard and tough due to the depositions of resins, tannins, gums, and so on. So this central hard, tough and darker region forms the heartwood. The next question relates with the chapter hormonal coordination. It's matched the following. We have column 1, column 2. In column 1, we have A, B, C, D, pituitary gland, thyroid, adrenal and pancreas. And in column 2, we have Graves' disease, diabetes mellitus, diabetes insipidus and Addison's disease. These are the options here, and this is the right answer. Okay. Now let us understand this in details. The very first thing that we can see the picture showing up the Graves' disease patient. Graves' disease, which is also known as toxic goiter, which is an autoimmune disease that affects the thyroid. It results in enlarged thyroid gland. Okay, so it shows the symptoms same as that of hyperthyroidism, like it includes irritability, muscle weakness, sleeping problems, a faster heartbeat, intolerance to heat, diarrhea, and weight loss. Other symptoms show thickening of the skin on the shins, that is called as the pretibial myxedema, eye bulging, very typical of Graves' ophthalmopathy. Then the second option, that is diabetes mellitus. Please remember, diabetes mellitus is caused due to deficiency of insulin hormone, and diabetes insipidus occurs because of deficiency of ADH hormone. Now, diabetes mellitus is a chronic condition in which there's an abnormally high level of sugar, that is glucose, in the blood. Because the insulin produced by the pancreas, it lowers the blood glucose. So, if at all insulin is produced in insufficient quantities or the body is not able to produce insulin, then this particular problem arises. Now, regarding diabetes insipidus, which is a very rare disorder and wherein a person's kidneys pass an abnormally large volume of urine, and we call it as insipid because insipid का मतलब होता है dilute and odorless. So, a person's body regulates the fluid by balancing the liquid intake and removing the extra fluid. Now, this is, as I said, it is caused by the deficiency of vasopressin, which is also called as ADH, that is antidiuretic hormone, which is produced by the hypothalamus but stored in the pituitary gland until it is needed. This regulates the level of water in our body by controlling the amount of urine our kidneys produce. So, when the level of water in our body decreases, the pituitary gland releases vasopressin, that is ADH, to conserve water and stop the production of urine. Then we have the next one, that is Addison's disease, a disorder in which the adrenal glands don't produce enough hormones. So specifically the adrenal glands produce insufficient amounts of the hormone cortisol and sometimes aldosterone too. Clear? So Addisonian crisis usko kaha jata hai, and that is what is it. The next question relates with the chapter biotechnology. It's a match the following. We have A, B, C, D as Bacillus thuringiensis, Thermus aquaticus, Agrobacterium tumefaciens, and Salmonella typhimurium. And 1, 2, 3, 4 are cloning vectors, construction of first rDNA molecule, that is recombinant DNA molecule. Third is DNA polymerase, and fourth is glycoproteins, and these are the options given. The correct answer is the first one, Thermus aquaticus. Bacillus thuringiensis being A. A can be related with cryproteins. Thermus aquaticus can be related with DNA polymerase. Then C, that is Agrobacterium tumefaciens, is a cloning vector. And D, Salmonella typhimurium, is the construction of the first rDNA molecule, that is recombinant DNA molecule. Now, Bacillus thuringiensis, you know, it's a gram-positive soil-dwelling bacterium, and it's a commonly used up as a biological pesticide. It occurs naturally in the gut of the caterpillars of various types of moths and butterflies, as well as on the leaf surfaces. Okay, now spores and crystalline insecticidal proteins produced by Bacillus thuringiensis have been used to control insect pests since 1920s and are often applied as liquid sprays. They are now used as specific insecticides under the trade names such as dipel and thuricide. Because of their specificity, these pesticides are regarded as eco-friendly. Okay, and they don't have really a negative effect on human beings or wildlife or pollinating agents and most of the other beneficial insects. And this is the reason why these are used up in organic farming. Now, what are cryproteins? It is a toxic crystal protein encoded by cry gene, which is CRY gene, which is toxic to certain insects, and this is produced by Bacillus thuringiensis. That's the reason A option can be related with fourth, that is cryproteins. Activation of cryprotein takes when ingested by the insects due to the alkaline pH of the gut. Now, the epithelial cells of the gut, they undergo lysis, and this leads to the death of the insects. Humans are taking advantage by incorporating Bt toxin gene in the crops to form the transgenic crops which are insect resistant. For example, you can say Bt cotton. इसके भी बहुत सारे questions अपने paper में है. Now let's know about Thermus aquaticus, option B. It is a species of bacteria that can tolerate high temperature, one of the several thermophilic bacteria which belongs to Deinococcus thermus group. It is a source of heat-resistant enzyme that is called as the Taq DNA polymerase, one of the most important enzymes in molecular biology because of its use in the PCR amplification technique. DNA polymerase was first used from Thermus aquaticus in 1976, and it is found to be thermostable at a temperature of 72 degrees, and it does not even denature at 95 degrees, and this is the reason why it is used. Now let's understand more about Agrobacterium tumefaciens. Its scientific name being Rhizobium radiobacter. Synonym is at times Agrobacterium radiobacter. It is a causing agent of crown gall disease. It involves formation of tumors in over 140 species of plants. This is a rod-shaped gram-negative bacterium. Okay. Now, these symptoms are caused by the insertion of a small segment of DNA that is called as the tDNA or transfer DNA, which is not to be confused with tRNA, that is transfer RNA. So this can be transferred from a plasmid into the plant cell, which is incorporated at a semi-random location into the plant genome. Now let's know about Salmonella typhimurium. It is a pathogenic gram-negative bacterium predominantly found in the intestinal lumen. Its toxicity is due to the lipopolysaccharide, you may be knowing the membrane, which protects the bacteria from the environment, which consists of O antigen, a polysaccharide core, and lipid A, something similar to Salmonella typhi, paratyphi, I mean to say. Okay, so this is it. It has been used to investigate its ability to deliver DNA to antigen presenting cells in order to produce cancer vaccines. So this is the reason why we relate it with construction of the first recombinant DNA molecule. The construction of the first recombinant DNA emerged from the linking of the gene encoding antibiotic resistance gene with the native plasmid of Salmonella typhimurium, and this was done by Stanley Cohen and Herbert Boyer in 1972. They isolated the antibiotic gene by cutting out a piece of DNA. Friends, thank you, and hang on for the other 80 questions as well. Hope you'll pass out with flying colors. Thank you so much.